Dear students, today we will discuss on diseases of guava and its management. Guava is an important fruit crop of subtropical countries. In India, it is grown almost in all the states. It is a hardy crop and is cultivated successfully even in neglected soils and is attacked by a large numbers of pathogens, mainly fungi. About 177 pathogens are reported to occur on various parts of the crop, including fruits, causing various diseases. The present topic will discuss on the symptoms and management of important guava diseases. So let's see the first important disease, that is wilt. The exact cause of the disease is still not fully understood, but the pathogens such as Fusarium oxysporum, Forma specialis, CD, Fusarium solani, Macrophomina fezulina, Rhizoctonia bataticola, Cephalosporium species and Gliocladium roseum are reported to cause wilting. Now let's see about the symptoms of the disease. Wilt is a pernicious disease and a curse to guava industry. First external symptom of the disease is the appearance of yellow coloration with slight curling of the leaves of the terminal branches. Plants at a later stage show unthriftiness with yellow to reddish discoloration of leaves. Subsequently, there is premature shedding of leaves. Some of the twigs become bare and fail to bring forth new leaves or flowers and eventually dry up. Fruits of the affected branches remain underdeveloped, hard and stony. Later, the entire plant defoliates and eventually dies. Next is the mode of spread of the disease. Thorough movement of plants containing sick soil in virgin areas. Short distance spread is by water. Root injury predisposes wilt disease. Next, let's see about the reason of severity. High rainfall during August or September, stagnation of water in guava field for long duration, temperatures ranges of 23 to 32 degrees centigrade with 76% relative humidity are conducive. Now let's see about the management of the disease. Proper sanitation in the orchard. Wilted trees should be uprooted, burned and trenches should be dug around the tree trunk. Manuring, interculture and irrigation enable them to withstand infection. The pits may be treated with formalin and covered for about 3 days, and transplanting should be done after 2 weeks. Organic manures, well cakes and lime also check the disease. Use of rootstocks resistant to wilt could be an alternative effective method for the control of disease. Cross of Pseudium malay and Pseudium guajava has been found free from wilt, and these material can be used as resistant rootstock. Biological control by Aspergillus niger strain AN17 is found effective. Multiply Aspergillus niger in FIM at the rate 5 kg per pit. Apply in the pits while planting new plants. In older plants, Aspergillus niger enriched FOM can be applied at the rate 10 kg per plant. So this is all about the wilt disease of guava. Next, see the another important disease, that is the anthracnose. It is caused by Gliosporium CD. Now coming to the symptoms of the anthracnose. First is dieback phase. The plant begins to die backwards from the top of a branch. Young shoots, leaves and fruits are readily attacked while they are still tender. The greenish color of the growing tip is changed to dark brown and later to black necrotic areas extending backwards, causing the dieback. Fruit and leaf infection phase. Fruit and leaf infection is generally seen in rainy season. Pinhead spots are first seen on unripe fruits, which gradually enlarge. Spots are dark brown in color, sunken, circular, and several spots coalesce to form bigger lesions. Unopened buds and flowers are also affected by disease which caused shedding. On leaves, the fungus causes necrotic lesions at the tip or on the margin. Next, about the mode of spread of the disease. By wind-borne spores available on dead leaves, twigs and mummified fruits in the orchard. Dense canopy is congenial for germinating of spore due to suitable moisture regime. Movement of planting material through infected foliage. Transportation of fruits from high disease pressure area without any treatment. Next is the reasons for severity. Closer planting without canopy management, lack of timely harvesting, availability of free water or rain encourages spore production and its dispersal around canopy. Now let's see about the management of the disease. Spray of body mixture, that is 3:3:50 or copper oxychloride at the rate 0.3% just after initiation of the disease. For post-harvest treatment, 20-minute dip in 500 ppm tetracycline is effective. Application of bioagents such as Streptosporangium pseudo-vulgare on fruits before emergence of the symptom. So this is all about the disease. Next is the canker. It is caused by Pathology CD. Now let's see the symptoms of the canker. The disease generally occur on green fruits and rarely on leaves. The first evidence of infection on fruit is the appearance of minute brown or rust color unbroken circular necrotic areas which in advanced stage of infection. Tears open the epidermis in the succinate manner. The infected fruits remain underdeveloped, become hard, malformed and mummified and drop. Now let's see the mode of the spread. The pathogen is primarily a wound parasite and avoids injury to fruits. Reason for severity of the disease. Germination of spores is maximum at 30 degrees centigrade and do not germinate below 15 degrees centigrade or above 40 degrees centigrade with relative humidity of 98%. Now let's see the management of the disease. Spread of the disease in early stage of infection is controlled by 3 to 4 sprays of 1% body mixture or lime sulfur at 15 days interval. So this is about the management of the disease. Next disease is the algal leaf and fruit spot. It is caused by Cephalourus virescens and Cephalourus parasiticus. Now let's see about the symptoms of the algal leaf and fruit spot. Algae infects immature guava leaves during early spring flush. Minute shallow brown velvety lesions appear on leaves, and as the disease progresses, the lesion enlarges to 2 to 3 mm in diameter. On leaves, the spot may vary from specks to big patches. They may be crowded or scattered. Leaf tips, margins, or areas near the mid-vein are most often infected. On immature fruits, the lesions are nearly black. As fruits enlarge, lesions get sunken. Cracks frequently develop on older blemishes as a result of enlargement of fruits. Lesions are usually smaller than leaf spots. They are darkish green to brown or black color. Now let's see how to manage the disease. Control of algae can be achieved by spraying of copper oxychloride at the rate 0.3% 3 to 4 times at an interval of 15 days when initial symptoms are noted. Next important disease is Phytophthora fruit rot. It is caused by Phytophthora parasitica, Phytophthora nicotianae variety parasitica, and Phytophthora citricola. Now let's see about the symptoms of the disease. The symptom starts at calyx discs of the fruit during rainy season. Affected area is covered with whitish cotton-like growth which develop very fast as the fruit matures, and pathogens is able to cover almost the entire surface within a period of 3 to 4 days during humid weather. The skin of fruits below the whitish cottony growth become little soft, turns light brown to dark brown, and emits a characteristic unpleasant smell. Ultimately, such fruits either remain intact or drop off from the tree. Now let's see about the mode of spread of the disease. Rain and wind are important for spread. The pathogen produces a great number of sporangia on the surface of diseased tissues, principally when the temperature is near 25 degrees centigrade, and this is an important source of inoculums in the development of epidemics. Drops of rain are necessary for the liberation of sporangia from the infected plant material or soil. Infection ceases when temperature is below 15 degrees centigrade or above 35 degrees centigrade. Now let's see reason for severity of the disease. Cool, wet environmental conditions with high soil moisture favor disease development. High humidity, temperature of 28 to 32 degrees centigrade, poorly drained soils, and injuries are important for initiation of disease. Close plantation is also one of the important factor for the disease. Next is the management of the disease. Dithan Z78 at the rate 0.2% or Ridomil or L80 0.2% or copper oxychloride at the rate 0.3% are found effective to control foliar infection. Soil drenching with copper oxychloride at the rate 0.3% or Ridomil or L80 at the rate 0.2%. Plant spacing and fertilizer regimes should be managed to avoid unnecessary dense plant canopy. Next important disease is the styler and rot. It is caused by Phomopsis cedi and Phomopsis destructum. Now let's see about the symptoms of the disease. The visible disease symptom is the discoloration of the region lying just below and adjoining the persistent calyx. Such area gradually increases in size and turn dark brown. Later the affected area becomes soft. Along with the discoloration of epicarp, the mesocarp tissue also shows discoloration and the diseased area is marked by pulpy and light brown color in contrast to the bright white color in the healthy area of the mesocarp. Next is the management of the disease. Spray copper oxychloride at the rate 0.3% or carbendazim or thiophanate methyl at the rate 0.1% before onset of winter fruiting. However, care should be taken that no spraying is done 15 days prior to harvesting. Avoid fruit injury. So this is about the management of the disease. Next important disease is the soft watery rot. It is caused by Botryodiplodia theobrome. Now let's see the symptoms of the disease. The infection starts as a brownish discoloration mostly at the stem end and it gradually proceeds downwards in an irregular wavy manner. Finally, the whole fruit may get involved. The decay takes the form of a soft, watery breakdown resulting from infection via wounds or through the stem end. Now let's see the management of the disease. Ensure careful handling in order to reduce the incidence of wounding. Captan and homeopathic drug arsenic oxide are found effective against the fungus. Application of Bacillus subtilis and Streptosporangium pseudo-vulgare on guava fruits have been found effective. Next important disease is the parasite. So it is a Loranthus species. Now let's see the symptoms of this parasite. The guava tree is the prey of many parasites and the most interesting of these are so-called phanerogamic parasites and epiphyses. The foliage infected host plant is sparse, reduced in size and its bearing capacity and quality of fruit is considerably lowered. Since the appearance of parasitic plant is quite distinct from the guava host, they can be easily distinguished in infected trees. The point at which the guava host is penetrated is usually characterized by swollen growth called burrs. The burrs help in the identification of sites at which the parasite has entered the host, an important feature while controlling these plants. Now coming to the management of these parasites. The affected branches should be cut sufficiently to eradicate the hosteria of the parasite. Spray of emulsion of diesel at the rate 30 to 40% in soap water is also found to be effective. So these are the all important diseases of guava. Coming to the conclusion of this topic, guava is affected by many diseases like wilt, canker and anthracnose. These diseases are of national importance and causes economic losses in guava. Timely application of control measures such as application of fungicide can minimize the foliar diseases. Soil-borne diseases can be reduced by soil solarization, growing resistant rootstock and also application of chemical and biological control agents.